In this feature presentation, we will describe the major features and chemical events during photosynthesis and respiration. This includes light reaction and dark reaction. We will also explain the importance of chlorophyll and other pigments. So do the major features and sequence during the chemical events of cellular respiration. And lastly, we will distinguish the major features of glycolysis, Krebs cycle, and electron transport chain. Now let's start with photosynthesis, or also known as the cycle of plants and how they make energy. For plants to perform photosynthesis, they require light energy from the sun, water, and carbon dioxide. Water is absorbed from the soil into the cells of roots. The water passes from the root system to the xylem vessels in the stem until it reaches the leaves. Carbon dioxide is absorbed from the atmosphere through pores in the leaves called stomata. The leaves also contain chloroplasts, which hold chlorophyll. The sun's energy is captured by the chlorophyll. Later on, we will explain what are the importance of chlorophyll, and so do the pigments.
Plants appear green because they reflect yellow and green wavelengths of light. Red and blue are absorbed by the pigments provide energy used. The light-independent reactions and the light-dependent reactions occur on the plants. So moving on, chloroplast, an organelle which contains chlorophyll. Photons from the sun are absorbed by the chlorophyll. Thylakoid, which is inside the chloroplast, is a phospholipid bilayer, which means there are hydrophobic and hydrophilic tails. It is stacked or arranged as a grana, and there is a membrane or fluid which is called the lumen, and then the stroma surrounds it. The light reactions of photosynthesis takes place in this. Light-dependent reactions are the first major set of processes in the photosynthesis. It converts light energy into chemical energy as ATP and NADPH. Photosystem 2 or plastoquinone oxidoreductase is the first of the four protein complexes that plants need for light-dependent reactions. It is also called PS2, and it is called as it is because it was discovered the second after the photosystem 1. When photons hit the chlorophyll, an electron molecule gets excited. The term excited means an electron gaining energy. All energized electrons proceed to an electron transport chain, transported to the cytochrome B6F complex. It is where energy from electron to pump a proton in the thylakoid. But since there are electrons lost in the PS2, it splits the H2O or the water. Oxidation of water molecule produces free electrons and oxygen itself. As electrons pass through the transport chain, the energy from the electron is used to pump hydrogen ion or protons from the stroma to the thylakoid, creating a concentration gradient. A concentration gradient powers a protein called ATP synthase, which phosphorylates ADP.
The ATP and NADPH produced during light-dependent reaction will be used to produce sugar in Calvin cycle during light-independent reaction. The Calvin cycle occurs in the stroma of chloroplasts. So first, these 6 black dots represent 6 carbon dioxide molecules and need to be combined with 6 RUBP or ribulose biphosphate. Each one of RUBP will attach to the 6 carbon dioxide molecules. As it fixes the carbon, it will produce unstable 6 carbon molecule. Each will break in half, producing 12 PGA. This cannot happen without RUBP carboxylase, which is an enzyme, as it helps on catalyzing 6 carbon dioxide attachment to 6 RUBP. RUBP carboxylase comes in, catalyzing the reaction. Going back to 12 PGA, it will use 12 ATP from light-dependent reaction, converting it into 12 ADP plus P. It will now then use 12 NADPH, converting it into 12 NADP plus, producing 12 PGAL. These 12 PGAL need to settle down its two molecules. 10 PGAL will remain. It will again use 6 ATP from light-dependent reaction, converting it into 6 ADP plus P. They will arrange themselves to produce the original 6 RUBP. Then, the two molecules here will now become a molecule of sugar, RC6H12O6.
Pigments are chemical compounds which reflect only certain wavelengths of visible light. This makes them appear colorful. Flowers, corals, and even animal skin contain pigments which give them their colors. More important than the reflection of light is the ability of pigments to absorb certain wavelengths. Photosynthesis in plants is dependent upon capturing light energy in the pigment chlorophyll, and in particular chlorophyll A. This chlorophyll resides mostly in the chloroplast and gives leaves their green color. The range of light absorption in leaves is extended by some accessory pigments such as the carotenoids, but does not cover the entire visible range because that would make the leaves black. Chlorophyll A is the primary pigment for photosynthesis in plants. It has the composition C55H72O5N4Mg. It exhibits a grass green visual color and absorption peaks at 430 nanometer and 662 nanometer. It occurs in all photosynthetic organisms except photosynthetic bacteria. It reflects green light strongly, so it appears green to us. Chlorophyll B has the composition C55H70O6N4Mg. The difference from chlorophyll A being the replacement of a methyl group with a CHO. It exhibits a blue-green visual color and absorption peaks at 453 nanometer and 642 nanometer. It occurs in all plants, green algae, and some prokaryotes. There is usually about half as much as chlorophyll B as the A variety in plants. It is not as abundant as chlorophyll A and probably evolved later. It helps increase the range of light a plant can use for energy. Carotenoids. This is a class of accessory pigments that occur in all photosynthetic organisms. They are completely hydrophobic or fat-soluble and exist in lipid membranes. Carotenoids absorb light maximally between 460 nanometers and 550 nanometers and appear red, orange, or yellow to us. The colors of the carotenoids are familiar as the fall colors of leaves. As the leaves lose their chlorophyll, the more persistent carotenoids give the pleasant reds, yellows, and oranges of the autumn foliage. Beta-carotene is the most important of the carotenoids. Lycopene, the color of tomatoes, is also a carotenoid. Beta-carotene is the most important of the carotenoids that serve as accessory pigments in photosynthesis. Measurement of the absorption of these pigments as a function of photosynthetic output makes it clear that the chlorophylls are the most important, but that beta-carotene contributes. Beta-carotene gives its color to the carrot and also to squash, bananas, and avocados. It also appears in autumn leaf colors. Lycopene is familiar as the red color in a tomato.
Cellular respiration is a metabolic pathway that breaks down glucose and produces ATP. The stages of cellular respiration include glycolysis, pyruvate oxidation, the citric acid or Krebs cycle, and oxidative phosphorylation. During cellular respiration, A glucose molecule is gradually broken down into carbon dioxide and water. Along the way, some ATP is produced directly in the reactions that transform glucose. The first step is the glycolysis. Glycolysis occurs in the presence of oxygen and is when glucose is broken down. This occurs in the cytoplasm. The result of glycolysis is two molecules of ATP, pyruvate, and NADH. Before the second main step begins, the pyruvate undergoes oxidation in the mitochondria and is converted into acetyl-CoA. NADH gains electrons and carbon is lost, which forms carbon dioxide, or also known as CO2. And the last main portion of cellular respiration is oxidative phosphorylation. Electrons from NADH and FADH2 are transferred through the membrane of the mitochondria. When hydrogen ions come back across the membrane, ATP is synthesized through what is called the ATP synthase complex.
Glycolysis is the metabolic process that serves as the foundation for both aerobic and anaerobic cellular respiration. In glycolysis, glucose is converted into pyruvate. The first step in glycolysis is the conversion of D-glucose into glucose 6-phosphate. The enzyme that catalyzes this reaction is hexokinase. The second reaction of glycolysis is the rearrangement of glucose 6-phosphate into fructose 6-phosphate or F6P by glucose phosphate isomerase, or also known as phosphoglucose isomerase. Phosphofructokinase, with magnesium as a cofactor, changes fructose 6-phosphate into fructose 1,6-biphosphate. Enzyme aldolase splits fructose 1,6-biphosphate into two sugars that are isomers of each other. These two sugars are dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. The enzyme thiophosphate isomerase rapidly interconverts the molecules dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate dehydrogenates and adds an inorganic phosphate to glyceraldehyde 3-phosphate, producing 1,3-bisphosphoglycerate. Phosphoglycerate kinase transfers a phosphate group from 1,3-bisphosphoglycerate to ADP to form ATP and 3-phosphoglycerate. The enzyme phosphoglyceromutase relocates the P from 3-phosphoglycerate from the third carbon to the second carbon to form 2-phosphoglycerate. The enzyme enolase removes a molecule of water from 2-phosphoglycerate to form phosphoenol pyruvic acid. The enzyme pyruvic kinase transfers a P from phosphoenol pyruvate to ADP to form pyruvic acid in ATP that results in step 10 or the last step.
The Krebs cycle chain, also known as the critic acid cycle, in which pyruvate is broken down into carbon dioxide. During the glycolysis, glucose is broken down into pyruvate. Pyruvate will be converted to acetyl once it passes inside the outer membrane of the mitochondria by the pyruvate dehydrogenase complex, which is an oxidative process wherein NADH and carbon dioxide are formed. So from 3 carbon ion molecules in a pyruvate, it will be 2. The Krebs cycle actually starts once CoA is combined to the acetyl for it to be attached to the 4-carbon acceptor molecule, which is the oxaloacetate, to form citrate. Citrate is a 6-carbon molecule which releases 2 of its carbon as carbon dioxide molecules in a pair of similar reactions, producing a molecule of NADH each time. The remaining 4-carbon molecule undergoes a series of additional reactions, first making an ATP molecule, or in some cells, a similar molecule called GTP. Then reducing the electron carried FAD to FADH2 and finally generating another NADH. This set of reactions regenerates the starting molecule oxaloacetate so the cycle can repeat. The remaining four. Overall, one turn of the critic acid cycle releases 2 carbon dioxide molecules and produces 3 NADH, 1 FADH2, and 1 ATP or GTP. The critic acid cycle goes around twice for each molecule of glucose that enters cellular respiration because there are 2 pyruvates and thus 2 acetyl CoAs made per glucose.
Hydrogen carries the net high-energy electrons to the electron transport chain. As the electrons move through the chain, they lose energy, which they are transferred to the electron carriers within the chain. The electron carriers use this energy to pump hydrogen ions from the matrix and into the intermembrane space. The accumulation of H plus ions in the intermembrane space creates an electrochemical gradient or a proton motive force. H plus ion return to the matrix via transmembrane enzyme ATP synthase. This diffusion of ions is called chemiosmosis. As the ions pass through the ATP synthase, they trigger a phosphorylation reaction which reduces ATP from ADP plus Pi. The de-energized electrons are removed from the chain by oxygen, allowing new high-energy electrons to enter the chain. Oxygens also bind matrix protons from the water. This maintains the hydrogen gradient by removing H plus ions from the matrix.